This is the handout you've been provided. I will be reading it in this document for those that are interested in listening to it being read. Again, as you go through, you're looking for evidence that supports whether or not HeLa cells should continue to be used for scientific research. I would recommend using two different colors of highlighter. One color for any information that supports they should be used, and the second color for information that supports that these cells should not continue to be used. Moving into the reading, medical researchers use laboratory-grown human cells to learn the intricacies of how cells work and test theories about the causes and treatment of diseases. The cell lines they need are immortal. They can grow indefinitely, be frozen for decades, divided into batches, and shared among scientists. In 1951, a scientist at Johns Hopkins Hospital in Baltimore, Maryland, created the first immortal human cell line with a tissue sample taken from a young black woman with cervical cancer. Those cells, called HeLa cells, quickly became invaluable to medical research, though their donor remained a mystery for decades. In her book, The Immortal Life of Henrietta Lacks, journalist Rebecca Skloot tracks down the story of the source of the amazing HeLa cells, Henrietta Lacks, and documents the cell line's impact on both modern medicine and the Lacks family. Who was Henrietta Lacks? She was a black tobacco farmer from Southern Virginia who got cervical cancer when she was 30. A doctor at Johns Hopkins took a piece of her tumor without telling her and sent it down the hall to a scientist there who had been trying to grow tissues in culture for decades without success. No one knows why, but her cells never died. Why are her cells so important? Henrietta's cells were the first immortal human cells ever grown in culture. They were essential to developing the polio vaccine. They went up in the first space missions to see what would happen to cells in zero gravity. Many scientific landmarks since then have used her cells, including cloning, gene mapping, and in vitro fertilization. There has been a lot of confusion over the years of the source of HeLa cells. Why? When the cells were taken, they were given the code name HeLa for the first two letters in Henrietta and Lacks. Today, anonymizing samples is a very important part of doing research on cells. But that wasn't something doctors worried about much in the 1950s, so they weren't terribly careful about her identity. When some members of the press got close to finding Henrietta's family, the researcher who grown the cells made up a pseudonym, Helen Lane, to throw the media off track. Other pseudonyms, like Helen Larson, eventually showed up too. Her real name didn't really leak out into the world until the 1970s. How did you first get interested in this story? I first learned about Henrietta in 1988. I was 16 and a student in a community college biology class. Everybody learns about these cells in basic biology, but what was unique about my situation was that my teacher actually knew Henrietta's real name and that she was black. But that's all he knew. The moment I heard about her, I became obsessed. Did she have any kids? What do they think about part of their mother being alive all these years after she died? Years later, when I started being interested in writing, one of the first stories I imagined myself writing was hers. But it wasn't until I went to grad school that I thought about trying to track down her family. How do you win the trust of Henrietta's family? Part of it was that I just wouldn't go away and was determined to tell the story. It took almost a year, even, to convince Henrietta's daughter, Deborah, to talk to me. I knew she was desperate to learn about her mother. So when I started doing my own research, I'd tell her everything I found. I went down to Clover, Virginia, where Henrietta was raised, and tracked down her cousins, then called Deborah and left these stories about Henrietta on her voicemail. Because part of what I was trying to convey to her was I wasn't hiding anything, that we could learn about her mother together. After a year, finally she said, "Fine, let's do this thing." When did her family find out about Henrietta's cells? 25 years after Henrietta died, a scientist discovered that many cell cultures thought to be from other tissue types, including breast and prostate cells, were in fact HeLa cells. It turned out that HeLa cells could float on dust particles in the air and travel on unwashed hands and contaminate other cultures. It became an enormous controversy. In the midst of that, one group of scientists tracked down Henrietta's relatives to take some samples with hopes that they could use the family's DNA to make a map of Henrietta's genes so they could tell which cell cultures were HeLa and which weren't. So to begin straightening out the contamination problem. So a postdoc called Henrietta's husband one day, but he had a third-grade education and didn't even know what a cell was. The way he understood the phone call was, "We've got your wife, she's alive in the laboratory. We've been doing research on her for 25 years, and now we have to test your kids to see if they have cancer," which wasn't what the researchers said at all. The scientists didn't know that the family didn't understand. From that point on, though, the family got sucked into this world of research they didn't understand. And the cells, in a sense, took over their lives. How did they do that? This was most true for Henrietta's daughter. Deborah never knew her mother. She was an infant when Henrietta died. She had always wanted to know who her mother was, but no one ever talked about Henrietta. So when Deborah found out that this part of her mother was still alive, she became desperate to understand what that meant. Did it hurt her mother when scientists injected her cells with viruses and toxins? Had scientists cloned her? And could those cells help scientists tell her about her mother, like what her favorite color was and if she liked to dance? Deborah's brothers, though, didn't think much about the cells until they found out there was money involved. HeLa cells were the first human biological materials ever bought and sold, which helped launch a multi-billion-dollar industry. When Deborah's brothers found out that people were selling vials of their mother's cells and that the family did not get any of the resulting money, they got very angry. Henrietta's family has lived in poverty most of their lives, and many of them can't afford health insurance. One of her sons was homeless and living on the streets of Baltimore, so the family launched a campaign to get some of what they felt they were owed financially. It consumed their lives in that way. What are the lessons from this book? For scientists, one of the lessons is that there are human beings behind every biological sample used in the laboratory. So much of science today revolves around using human biological tissue of some kind. For scientists, cells are often just like tubes or fruit flies. They're just inanimate tools that are always there in the lab. The people behind those samples often have their own thoughts and feelings about what should happen to their tissues, but they're usually left out of the equation. And for the rest of us, the story of HeLa cells and what happened with Henrietta has often been held up as an example of a racist white scientist doing something malicious to a black woman. But that's not accurate. The real story is much more subtle and complicated. What is very true about science is that there are human beings behind it, and sometimes even with the best of intentions, things go wrong. One of the things I don't want people to take from the story is the idea that the tissue culture is bad. So much of medicine today depends on tissue culture. HIV tests, many basic drugs, all of our vaccines, we would have none of that if it wasn't for scientists collecting cells from people and growing them. And the need for these cells is going to get greater, not less. Instead of saying we don't want that to happen, we just need to look at how it can happen in a way that everyone is okay with.